here, just one brake system. But that brake system composed of P16, P15, P18, P19. The late G1 phase controlled by cyclin ECDK2. And then they have all these CQ family to control that. They have control that target of cyclin CDK. Cyclin CDK actually in turn regulate an important protein involved in cell proliferation called RB, okay? Retinoblastoma protein, okay? RB is very important transcriptional regulator because RB, okay, was able to control E2F transcriptional activity, thereby control all the genes involved in G1 to S phase biological function. Okay. And then after that, the cell go to DNA synthesis. DNA synthesis, sometimes all these CDKs still play an important checkpoint. Just try to make sure the DNA synthesis, DNA duplication process is intact. If any problem happens, the cell will be able to stop it. Okay. The mitosis, go to mitosis, cyclin BCDK, will be able to stop the mitosis process because mitosis is also delicate. It's such a short time. The chromosome need to be condensed, chromosome need to be aligned, chromosome need to be equally separated into two daughter cells. Okay, you want to make sure that completely without any problem. Now we know some of the birth defect problems like Down syndrome, because of chromosome separation problem, and some of the cancer cells have the problem of causing cancer because they will be able to make use of the deregulation of the mitosis, okay, causing some of the cancer cells have an extra copy of the chromosome. So they will be able to have an advantage of cell proliferation or evade from immunosurveillance, evade from the apoptosis control. Okay? So these mitosis is such an important process. We will talk about that later. Okay? I think right now it's 50 minutes. We could have a short break, and then on we could go on to talk about how the cancer cell have the capability to inactivate all these important cell cycle break. Okay. Any questions you have? Okay. Use microphone, please.
Sorry, I don't remember. Did you mention about the 1433 sigma?
Yes.
So how the mechanism is?
Good. You are very inquisitive. Okay. The 1433 sigma, I didn't go into detail, but 1433 sigma also, just like P21, P27, they have CDK interaction motif. They will be able to interact with CDK. In the mechanism it does is sequester the CDK from going to the nucleus. So what 1433 does is, because the cyclic CDK need to go to the nucleus in order to be activated, and in order to do their biological activity. But 1433 sigma prevent CDK from going to the nucleus. So CDK could not go in, cyclin there is no function. So thereby inactivate cyclic CDK activity. So mechanism-wise, 1433 sigma is different from P21, the CQ family or the INK4 family. Good question, yeah.
And the second question is, so for controlling the cell cycle, we only have the negative regulators. So is there any activator in our?
Activator, as I mentioned, the positive regulator, that includes cyclin CDK. So for cancer, some of the cancer, they manage to have overexpression of cyclin E or cyclin D. Okay, So because of these, they manage to continue to maintain these protein stability. Because these cycling, why they get the name from, they come and go. When they finish their cycle, they get degraded, but then some of the cancer cell will be able to manage to keep these cycling from degradation. Okay? So could be because of this mechanism, or sometimes because the amplification of the cyclin E gene to make the cancer be able to push the cell cycle faster.
Sorry. So for the normal growth of human or any animal, is there any stage of our life that we need to push the cell cycle faster? Not cancer.
Well, definitely for example, your wound healing process, right? When you got hurt, you need your skin to heal faster. This process you need a cell proliferate fast, right? So indeed we do need that, and not only that, you grow hair, right? You need the cell proliferation, and your intestine system continue to be replaced. Your intestine system, the epithelial cell, go through the cell cycle proliferation, go through a differentiation process to make sure your epithelial cell to be able biological activity to keep on continue to absorb nutrition from the food you eat. Those that need to be cell proliferate. So that's the reason when the chemotherapy drug, when we apply to patient, we stop the cell cycle. Usually the patient hair loss, the patient has what? Diarrhea, because you're killing all these epithelial cells. And these epithelial cells in the intestine, they will not be able to absorb more nutrition, so you have diarrhea. Okay? So inside our body, we have certain type of cell continue proliferated because we need to replace using the new generation of cell to replace the old cell. Okay? So the hair loss, because the chemotherapeutic drug usually target the cancer cells, preventing them from cell proliferation. But they could not differentiate normal cell from cancer cell. That's the reason cancer patient lost hair, cancer patient has diarrhea.
Thank you.
I have a question about the P protein 27.
Yes.
I don't understand what is the goal of the P27 purification.
Purification.
Yes.
Why we need to do that?
Yes. And what's and the goal of purification of P27?
The goal of P27
Yes.
purification.
Yes,
Okay.
I don't understand.
Okay. So I just want to tell you, at that time, before P27 was characterized, we don't know what protein could regulate cyclic CDK activity. So at that time, just assume you don't know the P27 is there. Okay? So we need to understand what regulates cyclic CDK. So we're using the cell cycle arrested, assuming there's a CDK inhibitor there. And then we go through the process. The cartoon showing P27 there is after we identify the protein and then identify the gene, we make the migration, we run a gel and then it's located 27 kilodalton. So we name it the P27. So the goal of doing that process is to understand what protein bind to cyclic CDK, and the P27 was identified. Okay. Good. Yes?
I have a question. Has anyone tried to knock out one cyclin plus one CDK? Yeah.
Good question. Knock out both cyclic and
And
CDK?
see to resist this independently or dependent.
Yeah, because you only knock out one already have some problem, right? So for example, if you knock out CDK2, CDC2 already a problem. So this question have to be go a little bit detailed. Say which cyclin you want to knock out and which CDK you want to knock out at the same time. To my knowledge, maybe people have done that, but it also depends on what question you really want to ask.
I mean, just want to knock out one cyclin and one CDK, which are not typically together. So it's like a CDK 4 plus cyclin E.
Okay, I think that you have a good thought, but you have to consider the practical reason. Okay, you knock out cyclin, the cyclin may be replaced by other cyclin already, right? And you want to knock out another CDK, that CDK may be also replaced by other CDK. So you have to be carefully design which cyclin CDK in the same time you want to knock out at the same time. And so I think that you have a good thought about, doing all these sort of try to address certain ideas, but you need to make it as much more practical and understand the real questions you really want to address.
I mean, has anyone tried that?
Must be, but I don't think I have seen the paper published one cyclin CDK. Sometimes they use in the cell system the yeast system or the Drosophila system or the fruit fly system to do the study. Yeah.
Thank you.
Excuse me, Professor, regarding the histogram generated by the FACS system,
Yes.
I just want to ask, maybe I missed that idea, but how can you discriminate between the G1, the S, and the G2 cells based on that histogram?
Okay, good question. I think it is a research tool. The DNA histogram was actually stamped by a dye, okay, that could recognize the DNA. Usually we're using propylium iodide. So these are a DNA intercalating agent. They will bind to the DNA, and it depends on DNA content. The 2N and 4N. The 2N bind less, 4N will bind more, right? So when the cell pass through, there's a detector in the FACS analysis. They will based on the fluorescence, how strong the fluorescence of the propylium iodide bind to the DNA content, and that will allow you to know this is cell has 2N content, because once it pass through, you see, because the content is based on the nanometer of the fluorescence. I think the intensity of the fluorescence. So be able to tell. So 2N cell go to has certain fluorescent release. 4N content cell will release almost double the previous one. So then you could tell 2N and 4N. Okay.
Thank you.
Sure.
Sorry, teacher, I'm so curious with the checkpoint because in every gate, should they gate 1 and gate 0, we have a checkpoint. And could you tell me what is the checkpoint? Maybe what compose it?
Okay. You're talking about some of the last slide?
Yes.
The checkpoint? Okay. The checkpoint definition is just to make sure the cell is ready for the next step. Okay. So when the cell receives the signal, okay, I need to go to the second step. For example, the normal cell, you want to start doing duplication. The checkpoint makes sure the cell is ready for that. Okay, so the RB function is important checkpoint. The checkpoint gate there. Okay, when you don't have cyclin CDK activation, the RB is stopping there. RB is not phosphorylated. RB prevents E2F activity, transcription activity. But once cyclin CDK, because the G1 cyclin CDK become activated, the cell know I'm ready to go to the next step. Okay, so it will phosphorate on RB. RB got phosphorated and release the E2F, the transcription factor. This transcription factor, transcription activate many genes involved in DNA synthesis, involved in the G1S progression. Okay, so that restriction point is very important to let the cell go. Yeah. Okay, now you're ready to go to the next step. Okay. Another restriction point. But some of the restriction point also happen in mitosis. For example, we will talk about the mitotic checkpoint. Okay. The spindle fiber from a cell has been importantly attached to the centromere of the chromosome. Okay. Without proper attachment, the chromosome not supposed to be separated. Okay. So that checkpoint is very important. We'll talk about those so-called spindle checkpoint. Okay. So our cell cycle machinery do a wonderful job to make sure we are normal. Okay. If they don't do the job, then we will have trouble. Okay. So that's the reason, I think it's important for all of you who have studied biology, study biochemistry, you should pay attention to the cell cycle proliferation process. Okay. More questions? I'm very glad to hear a lot of questions. It's great. Keep that interacting going. Yeah, do you have a question?
Thank you for your lecture. And the first slide you mentioned about 8 kind of cycling and 13 about CDK. But in the lecture you just mentioned about 4 kinds of CDK. I don't know how about the others.
Okay.
Are they important?
Yeah, the others are important as well. But I didn't today focus more about cell cycle machinery. Other CDK, they do have cell cycle proliferation, but not so much directly. Some of the cycling CDK, for example, CDK8 or CDK9, they are involving the so-called RNA polymerase in C-tail phosphorylation event to make sure the polymerase has certain activity. So it's not so much directly involved in the RB regulation. Okay. So indeed they do have an important biological function, but this is only serve as an initiation point for you to understand more about cell cycle. Okay. Any more questions? Raise your hand. I think there's no stupid question. You should keep in mind. Any question deserve to satisfy your curiosity.
Sure. Thank you for your presentation.
Yeah.
I have one question that you talking about the kinase and the phosphatase, and you said that it will be activated or deactivated when it is phosphorylated. So we will control this by phosphoryl or the phosphorylation. So we will control the ATP over the AMP ratio, or control other enzyme for phosphorylation or phosphorylation.
Yeah, good. That's a good question. I think I didn't go into detail talk about those phosphorylation event or dephosphorylation event. Indeed, some of the important dephosphorylation event, for example, 12, 14, 15, there's a phosphatase has been characterized involved in this process called CDC25. Okay, this important phosphatase was able to dephosphorate the inhibitory phosphorylation site. I didn't talk much about this. Again, the cell cycle. We could talk about all the lecture about cell cycle, but I just point out that quickly. But if you're interested, the CDC25 is the phosphatase involved in that dephosphorylation event. And then the Sterling 160 has a kinase involved, okay, to catalyze the phosphorylation event on Sterling 160. So you see, even such a tiny kinase already has so many regulation events there. And then we should really appreciate what has been delicately control all these processes. The more I think I want to share with you, the more I study biology, I think we need to be more humble. Our little cell is like a little universe. Okay. Here we live on the earth, we think the earth is only part of a huge, gigantic universe, right? There's a galaxy, and outside of these galaxies, there's another one, there's another one. But cell is exactly the same situation. We know very little about it. Even these days, we have the Human Genome Project being completed. We still have so much to learn, okay? And I encourage everybody to be able to find your effort to continue to learn all these cell cycle regulatory process. It's so amazing, and then I'm sure the more you learn, you find, wow, this is there, that is there, and then and continue to develop your curiosity. I think that's the basic element for you to become a good researcher. Okay, and I have seen quite a few. You have a question, meaning that you developing that important curiosity, and that's a basic element to become a good researcher. Okay? And I really encourage you to continue to do so. Next question there. Okay. Hey.
You see me?
Yeah.
Thank you very much for your lecture. I have just one question for you. I want to ask about what control the time bound between phases in the cell cycle. I mean, there are different cells, different tissues in our body. Do all cells in the body have the same time generation? And in that case, what control the long? I mean, there are any cells that have shorter end phase or longer synthesis phase, or something like that.
Good question. Everybody understand the question, right? Okay. She was asking about our different tissue in our body has a different cell cycle regulation. Indeed, there are. In general, for a cell to complete a cell cycle, take about how long? Anybody know the answer? For a cell to become two daughter cells, how long does it take? Anybody know the answer? Or you could guess.
6 hours.
Come on, someone give me an answer.
6 hours.
Why don't you see if you know?
Maybe 1 day.
His answer is 1 day. That is the correct answer. Very nice. Okay. Okay, let me ask you another question. For a bacteria to become 2 daughter cells, how long does it take? Say E. coli. I hear some answer, but anybody shout?
1 hour.
Could you make the answer? How long?
Maybe 3 hours.
3 hours. He says 3 hours. Anybody have other answer? 20 minutes. Okay. Anybody have other answer? Okay. She's correct. Very close to 20 to 30 minutes for E. coli from 1 cell to become 2 daughter cells. But for human, for our mammalian cell, usually take about 24 hours to finish the whole cell cycle. Yeah, according to all these cell cycle clock, several phases only have short time. For example, the mitosis phase took about 30 to 1 hour to complete. Okay, so within such a short time, the cell, the chromosome, the DNA need to be condensed, need to be aligned, and then separate and done. Okay, so everything was doing such a short time. But the E. coli, during the duplication, only 20 minutes because the E. coli is, we call it prokaryote. It doesn't have the cell memory. It's only DNA duplication. Okay, 20 minutes. So that's the reason when you have a spoiled food, okay, have an E. coli contamination, you could imagine if that's original 100 E. coli there, and then 100 times 2, and then square, then you know how many E. coli will be after one day, right? So that's duplicate fast. So the answer for that, as I just mentioned, the cell proliferation, different cell, different type of tissue, they have different cell cycling time, okay? And in terms of cancer, some of the cancer cells do have the faster cell cycle progression. So their cycling time could be reduced from 24 hours to 18 hours. So you can see some of the people, hey, the guy who developed cancer, and then 3 months or 4 months later, you see the tumor grow so fast already. Okay. It's not overnight event, but you have an idea that cell cycle proliferation could be accelerated compared to normal cells.